And so now let's move to Hallie Randolph. And Hallie is a PhD at Luis Barreiro's lab at the University of Chicago. And she combines genomics with immunology to understand how transcriptional and genetic variation influence differences in the immune response across individuals. And the title of her talk today is Genetic Ancestry Effects on the Response to Viral Infection Are Pervasive but Cell Type Specific. So Hallie, feel free to share your screen and start when you are ready.
Sure. Can everyone see slides? All right,
yes,
great.
That's fine. That's fine.
Great. All right. Hi, everyone. Thanks for being here and thanks for that introduction. Today, I'm going to be talking about a project that involves characterizing the effects of genetic ancestry and genetic variation on the response to viral infection. And this work was recently published at the end of last year in Science. So you can also go and check it out there, of course. So about 60,000 years ago, modern humans migrated out of Africa. And during this process, they faced variable selection pressures, including many different pathogenic environments. Here, this map is showing what we know about the historical geographic distribution of pathogen abundance and diversity during the peopling of the world. And what really stands out is that pathogen exposure across populations varies pretty substantially. Specifically, it's thought that the geographic stratification of exposure to viruses was quite strong between populations. And we now know that in particular, RNA viruses have imposed very strong selection pressures on the human genome historically. Because of this, it's been hypothesized that variation in viral mediated selection pressures between different human populations in the past might have driven the divergence of immune responses among modern day human populations. There's a good amount of evidence to suggest that genetics, at least partially, plays an important role in explaining population variation in the response to influenza. There have been a few studies explicitly showing that regulatory variants control between individual differences in gene expression following infection with influenza A virus, which for the rest of this talk, I'm going to refer to as IAV. And this includes 2 studies performed in purified populations of immune cells that show that hundreds of cis-acting and a few trans-acting variants are associated with the transcriptional immune response to IAV. It's also been shown that following influenza infection, about 1/3 of genes display differences in expression that are correlated with genetic ancestry in monocytes derived from African and European individuals. To date, these and other immune response QTL studies all focus on a single isolated immune cell type, which means that any cell type specific genetic effects that might mediate response variation would be missed. So to address this gap, we used single cell RNA sequencing to profile the transcriptomes of peripheral blood mononuclear cells, or PBMCs, responding to IAV infection. And so I'm going to break this down into 3 parts that sort of mirror the structure of the paper. The first just being an overview of the study design and some of the main differences in response to IAV across cell types. Then I'm going to talk about population differences in response to IAV and the role that genetic variation plays in shaping these response differences. And then finally, I'll talk about how population variation may influence immune gene regulation more generally, using the response to SARS-CoV-2 as an example. And so first, just an overview of the study. We collected PBMCs from a cohort of 90 individuals, 45 which self-identify as European American and 45 as African American, and infected them with the pandemic H1N1 influenza A strain Calo 409, or we exposed them to mock condition media for 6 hours. Then after 6 hours of infection, I collected these samples for single cell RNA sequencing, which was done using the 10x workflow, and we also collected DNA for genotyping. In total, we collected around 235,000 high-quality single-cell transcriptomes across all individuals and conditions after QC filtering. And here I'm showing a UMAP that includes all of these cells, where you can see that 5 main clusters pop out, which correspond to the 5 major cell types in PBMCs. So these include the CD4 and CD8 T cells, the NK cells, the B cells, and the monocytes. There are also a few other rarer cell types, but the focus of this paper was really these 5 main ones. We first wanted to look at the overall signature of IAV infection, and we did this by collapsing the single cell gene expression values into pseudobulk estimates. We then identified genes that significantly alter their expression levels in response to IAV in each cell type. And you can see that out of the cell types that we considered, monocytes are the most responsive to infection, with almost 40% of their transcriptome being remodeled after exposure to IAV. We also looked at the level of intracellular IAV transcripts, which are influenza-derived transcripts that are generated and processed by infected host cells, and found that monocytes by far have the highest viral transcript levels, suggesting that these cells are either the most susceptible to infection, the most permissive to viral replication once the virus is actually inside the cell, or some combination of both of those factors. And so taken together, this data shows that monocytes are really the cell type that's most affected by IAV infection. And this is in line with other observations that have been noted in the literature. Now I'm going to switch gears to talk about population level differences in response to IAV, and also the contribution of cis regulatory genetic variation to these differences. So we also collected low pass whole genome sequencing for the same individuals, which was used to estimate the proportion of African and European admixture for each person that we included in the study. And in this structure plot, you can see the breakdown of those proportions for each individual. Most importantly, this quantitative admixture proportion is what's used in all of the downstream models to estimate the effects of genetic ancestry on gene expression. We first identified genes for which expression levels are correlated with genetic ancestry estimates. And these genes are what we term population differentially expressed, or POPDE genes. Across cell types and conditions, we identify about 2,000 POPDE genes, which corresponds to 29% of the genes that we tested, which shows that POPDE effects are common. But if we look at POPDE gene sharing across cell types, we found that the majority of POPDE genes were only identified in 1 or 2 cell types, suggesting that genetic ancestry effects on gene expression, although common, are also highly cell type specific. We then wanted to identify the functional pathways most closely associated with genetic ancestry. And to do this, we performed enrichment analysis using the hallmark gene sets. Before infection, we identified significant enrichments among multiple immune-related pathways, including the interferon alpha and gamma response pathways, among genes more highly expressed at baseline in individuals with a greater proportion of African ancestry. However, after infection, this pattern reverses, and we actually see an enrichment of type 1 and 2 interferon pathway activation in genes that are more highly expressed in individuals with a greater proportion of European ancestry. And so this data suggests that African ancestry individuals tend to have higher levels of interferon activation at baseline, but that this interferon pathway activity shifts to being higher in European ancestry individuals when the cells are actually exposed to the virus. To better characterize this shift, we constructed a per individual score of interferon signaling activity, which we termed the interferon score. And this was done by calculating the average mean centered expression of genes belonging to the hallmark interferon alpha and gamma gene sets. We then asked whether this score correlated with quantitative genetic ancestry estimates, and found that at baseline, there isn't any relationship. However, if we look at this following infection, we see that increased European ancestry strongly correlates with a higher interferon score across cell types. We then looked at the correlation between type 1 to interferon response magnitude and the level of IAV transcripts measured in PBMCs, and found that a stronger interferon response is associated with more intracellular IAV transcripts. And then in line with the stronger transcriptional interferon response that we detect in European individuals, we also see that individuals with increased European ancestry display higher levels of intracellular IAV transcript expression. Next, we wanted to functionally validate some of these findings, and to do this, I infected PBMCs from the 20 individuals with the strongest and weakest transcriptional interferon responses with IAV. And then we collected secreted cytokine measurements across 8 time points over 48 hours, as well as viral titer measurements at later time points. We found that high responders produced significantly more secreted interferon alpha and beta compared to low responders beginning at about 12 hours post-infection. And this is an effect that's actually exacerbated over time. We also found that viral titers quantified from supernatant 24 and 48 hours post-infection were significantly reduced in high responders compared to low responders, suggesting that individuals that are better able to mount type 1 interferon responses shortly after infection also display a greater capacity to actually limit productive viral replication at later time points. And so our working model shown here, from where our cohort level data, we know that European ancestry individuals show both a stronger transcriptional interferon response as well as higher levels of IAV transcripts at 6 hours post-infection. And then from our functional validation experiments, we know that high responders secrete significantly more interferons throughout time and show higher IAV transcript levels early after infection, which inverts at later time points. And so using these high responders as a proxy for European ancestry individuals more generally, this suggests that increased European ancestry may be associated with better viral control. Next, to assess the contribution of genetic variation to genetic ancestry-associated differences in the response to IAV, we mapped cis-QTL in both the mock and the IAV-infected samples. We identify about 2,200 genes that were associated with at least one cis-QTL across all cell types and conditions tested. And within cell type, on average, we found about 18% of E genes that were only detected in one condition. And one example is this variant in the interferon stimulated gene OIS1 that only shows a genetic effect following IAV infection. So that's that bottom panel here. We then wanted to ask whether genes with an eQTL were likely to be differentiated by genetic ancestry. And we found that E genes are strongly enriched for POP-DE genes and are about 3 to 6-fold more likely to be classified as POP-DE genes than expected by chance, suggesting that population differences in expression are likely to have a substantial genetic component. To formally measure the genetic contribution to population differences in expression, I calculated the correlation between our observed genetic ancestry effect and a predicted genetic ancestry effect that's based on the effect size of the top cis-QTL per E gene and the genotype for this SNP across individuals. These values were strongly correlated with the predicted differences in expression on average, explaining 53% of the variance in the observed genetic ancestry effects, which indicates that among POP-DE genes that have an eQTL, on average, over half of the population differences in expression are explained by cis-regulatory variation alone. And this means that there really is a considerable genetic component that's actually driving these differences. So then finally, we were interested in asking how variation in immune gene regulation might influence immune responses to RNA viruses more generally. Specifically, in the United States, health disparities in viral susceptibility and morbidity are pervasive. For example, the CDC estimates a 79% higher rate of influenza-related hospitalizations for Black versus white Americans, and similar health inequalities have been a defining feature of the COVID-19 pandemic, with racial and ethnic minorities dying at much higher rates than white Americans. And while a substantial proportion of these differences in morbidity and mortality can be attributed to health inequities due to structural and social determinants, we wanted to test whether variation in the immune response itself might either interact with or compound these existing health disparities. We hypothesized that if the ancestry-associated differences in expression identified in our in vitro infection model were to influence susceptibility, those genes should be enriched among genes associated with COVID-19 disease severity in vivo. And so to test this, we reanalyzed a large single-cell RNA-seq dataset collected from PBMCs of COVID-19 patients with varying degrees of severity. And in these patients, severity was measured by a metric called the World Health Organization ordinal scale, where lower scores indicate milder disease. Using this COVID patient cohort, we first identified genes with expression levels correlated with disease severity and found that monocytes by far have the greatest number of these severity-associated genes. We next asked if these genes tended to be ones that we identified as differentially expressed between European and African ancestry individuals in our data, and found that 23% of these severity-associated genes are POP-DE in our influenza dataset. And we also found that severity-associated genes are about 2 times more likely to be POP-DE genes compared to genome-wide expectations, meaning that we do see an enrichment of genes differentially expressed by genetic ancestry among these COVID severity-associated genes. And so taken together, this suggests that genetic ancestry-associated variation in immune responses might at least partially underlie some of the differences in COVID-19 susceptibility. Looking at a few examples of genes that have some of the strongest severity and POP-DE effects, we see that one set of genes stands out, and these are the S100 family members, which encode the S100 proteins. And one of the main functions of these proteins is to act as regulators of inflammation. So we identify S100A4, 6, and 8 as genes that are all strongly positively associated with severity, and also as genes that are significantly differentially expressed by genetic ancestry in monocytes. These genes have previously been shown to be associated with COVID severity and also patient improvement. And an independent paper showed that S100A, 8, and 9 are systemically upregulated in severe late-stage COVID-19 patients across different immune cell types. And these authors go on to hypothesize that this might actually be what's triggering cytokine storms in these patients. So then finally, we found that genes associated with increased COVID severity tended to be more highly expressed in monocytes of European ancestry individuals. And we see this both prior to infection and shortly after infection with IAV, which is the data that I'm showing here. But even though we see this trend, it's important to note that this observation alone does not indicate that European ancestry individuals are at increased risk for more severe COVID-19 outcomes, because here we've identified a transcriptional signature that's associated with severity in patients with an active SARS-CoV-2 infection. And while we know that this signature shows higher expression in European ancestry individuals, what we don't know is how this transcriptional signature is ultimately linked with outcome, where you can imagine having a stronger immune response early might be beneficial and lead to a milder course of disease, or it could be harmful and lead to a more severe disease outcome. And so what we now need are studies that find susceptibility signatures prior to infection to really properly address the question of directionality, considering variation in susceptibility. And with that, I'd like to thank my lab, especially Luis, for putting up with me and also being a really great sounding board throughout this project. Also Mari and Joao, who directly contributed to this work, as well as our collaborators, in particular the folks in Ryan Langlois' lab, who were super helpful with some of the more virology related aspects of the project. And I'd also like to thank my funding sources, and of course the organizers for giving me the opportunity to present this. Yeah, and I would be happy to take questions.
Okay, great. Thanks again for the very exciting talk. It looks like a lot of work that you had accomplished here. So we already have one question in the Q&A from Mike, and he's asking, what do you think is the mechanism that you found here? So do host variants directly increase interferon responsiveness to the same viral load? Or do the host variants allow stronger viral replication, which then indirectly triggers stronger interferon response?
Yeah, so that's something that we discussed a lot. And basically the answer is we're not sure. So I think we would really need to go back and do some more experimental tests to really get at that answer. It is worth noting, though, that the interferon signature that we see in the paper, we don't have very strong evidence to suggest that that's due to cisgenetic regulation. And so, of course, here we're not able to look at trans regulatory stuff just because, it's 90 individuals. We don't have the sample size to do that. But because we don't have very strong evidence that it is even like cisgenetically driven, one thing that we're wondering is, is there an environmental trigger that is actually correlated, for example, with genetic ancestry that is maybe driving that signature? And again, like that's not something given the metadata that we have on these individuals, unfortunately, that's not something that we were able to explicitly measure, but we are wondering if, that interferon signature is due to some environmental effect.
Great, thank you. So one of the questions I had, and it might be a bit naive since I don't know very much about infections or immunology, but what I was wondering is how these might change if you included in your culture that you infected some of like the actual target cells for the influenza virus. So when you spoke about COVID, like I'm not certain like that the virus would be infecting monocytes, for example, like maybe eventually, but its primary target would be
epithelial
cells in the respiratory tract.
Right, and respiratory,
and if you had co-cultured with that, might that have changed how these cells had responded to the infection? Rather than infecting the immune cells, infected the target cells and then maybe added the immune cells, if you've thought about that at all.
Yeah, it's definitely something that we've thought about. It's a really good question. The answer is I'm not really sure. It might change some of the conclusions. I think it's interesting to think about, like we talked about it a lot, a similar sort of thing in the context of, of course we're infecting PBMCs, but T and B cells historically, they don't tend to get infected by the virus. It really is like the monocytes and some of the NK cells that get infected. And also, of course, it's such a short time point post-infection. So we're looking here, at least for the big cohort level data, 6 hours post-infection. So even though we are throwing virus at T, B, NK monocytes, the ones that are really going to be responding truly in like a receptor dependent manner, like would be the innate immune cells, right? So it's really the NK cells and the monocytes that are sort of driving a lot of these signals. And in 6 hours, you're not really getting like a BCR and TCR dependent activation of those cells. So I think that's also something to think about. Like if we were to extend the time point, potentially how would some of these results maybe change? But it's not something that we did. But it's something that's, I think, really a good question to think about.
One additional question. So you had shown that with influenza A infection, that those with greater European ancestry can clear that virus more effectively. And at the very beginning, you had shown that a map showing basically pathogen distribution. So does in like the history of human travel and that sort of thing, were people of more European descent more exposed to influenza A? And that's why that they would mount a unique response?
I think, I think that, yes, probably were, like, European individuals did tend to be more exposed to influenza A historically, but I think jumping to that being the reason that this is something that we see is like a really hard thing to do. I think the hypothesis that this is probably driven by pathogen mediated selection is interesting, but again, like specifically for that interferon response signature, we don't, again, have a super good amount of evidence to suggest that that's cisgenetic. So it's likely, it's possible that that's not even driven by something like that was due to natural selection. It may just be, again, some environmental factor that's correlated with genetic ancestry, and that environmental factor is what's causing that to happen.
So we have another question in the Q&A. So Dan Holmes has asked, how uniform is the increase in interferon expression within monocytes? So are there low and high responding cells within one individual? And does that skewness change between people with different ancestries?
Yeah, that's a really good question. So this is something that we didn't really talk about in the paper at all, mostly because we were really focused on between individual variation. I did look at it briefly, and we do definitely see a variation in the levels of interferon expression, for example, in different monocytes within individual. I don't exactly remember how severe that was. And then as for the question of, whether that differs between ancestries, it's not something that I remember looking at that very specific analysis, and we don't see huge differences in that, at least at those single cell levels. But when we do the pseudo bulk modeling, that is something that stands out.
Okay, well, that looks like it's it for the Q&A. So thank you both, Haley and Andre, for taking the time to talk today. Congratulations to Andre on the Nature paper, and congratulations to Haley on this science publication. So our next seminar is scheduled for April 5th. So we hope to see all of you there. Thank you again for stopping by today.